So that's the title of my presentation, Extremophiles and Space Exploration. So I tried to put everything within 1 hour. Let's see if I can make it. Yeah, feel free to either stop me and ask questions or save your questions to the end. It doesn't matter. So I work for Blue Marble Space Institute of Science, which is a research institute that has a cooperative agreement with NASA. That's the same institution that Tomasz is also part of. So we are basically a research institute that manages grants, and also we have different programs, research programs and opportunities for young researchers, like the Young Scientist Program. And I'm going to talk more about that in the end of my presentation. So we are all enthusiastic about space sciences and astrobiology, and we have people from different backgrounds, geologists and oceanographers, biologists and chemists and so on. And we are proud to have Carl Pitcher, the former NASA Astrobiology Institute director, as part of our members. And here's Tomasz, of course. A lot of the research developed by Blue Marble researchers is related to many different types of studies of the environment, which includes extremophiles. So extremophiles, as you know, are organisms adapted or tolerant to different extremes, environmental extremes. And some people think that this is an anthropocentric concept, but it's actually not in many cases, because some of these places are very close to what is possible from the physical chemical point of view, in terms of the stability of molecules. For example, high temperature and high pressure, the organic molecules are very close to being destabilized, and still we are able to find organisms thriving in these environments. And we can think of many different types of parameters, environmental parameters, that defines different categories of extremophiles. And most of the extremophiles organisms are actually microorganisms, because that's what happens in nature. And even in our body, the vast majority of cells in our body is composed of microorganisms. And you can think of any type of environment. You can find a lot of microorganisms, much more than higher organisms, thriving in any type of environments, including the extreme environments. So microorganisms dominate all the different types of environments that you can think of. And I had the opportunity to participate in different projects at NASA. Here is an expedition to Mono Lake in California, very high pH, very alkaline environment. And we were isolating algae that were able to grow there. And here in the other extreme of the pH scale, very acidic environment in an iron mine in Colorado. Sorry, this is in Portuguese. It's an iron mine in Colorado. Very high not very high, but high altitude environment. It's a stream full of acidophiles, organisms adapted to very acidic conditions. And here again, the objective was to isolate algae that were able to grow at this extreme pH. And here is the main area of my research, which is studying deserts, microorganisms that can live in deserts and are exposed to radiation and are resistant to desiccation, and so are also tend to be resistant to radiation. And we will see why this is the case. So for the past 10 years, as I said, I've been collaborating in different projects at Lynn Rothschild's lab at NASA Ames. And as you can see, she has a very diverse portfolio of research, from biomining using microorganisms to do mining, precipitation of copper and other metals, biomaterials and bioprinting, synthetic biology of Earth. That's the Eucropis mission that I'm going to talk more about that. Human exploration using cyanobacteria to do in situ resource utilization. Cell-free expression systems. There's a collaboration with Keita Damala from the University of Minnesota in Minneapolis. Origin of life research, that's more related to what Tomasz is doing. Halophiles, and my research is more related to the limits of life, particularly extremophiles and radiation-resistant microorganisms. We also have engineers in our lab to do miniaturization of devices to do different objectives. Also search for life, and the iGEM competition, which is the International Genetically Engineered Machines competitions, very, very broad set of ideas that turned out to be turned into research projects in the lab. So the idea is students from all over the world go to a specific lab and develop an idea. It's a very, very early stage idea, and they do research in the lab for 3 months. Our lab receives students from Stanford University and from Brown University and a couple others institutions that also collaborate. And we had, along the years, many, many different projects like a biodrone, a bioballoon, self-folding origami, and Eucropis, mycotecture, astropharmacy. It's a lot of different research. And biowires. The idea here is to use a DNA molecule to incorporate metals, the first one being silver, and then use that as a wire, electro wire. So, as I said, my topic of research is limits to life, particularly radiation. And I also did a lot of developing this Eucropis mission that I'm going to talk more later. And all of these topics, they fall one way or another into astrobiology. And this is a document that was published in 2015. It's an astrobiology strategy. I think there's a new one coming up this year about astrobiology. It's basically what NASA is going to fund in the next 10 years. And I love this cover because it shows the extreme environments on Earth, and then a few planets in our solar systems, our planetary bodies in our solar system, and also the Milky Way galaxy at the background, and also a little comet here, showing all the different aspects of the possibility of finding life elsewhere through astrobiology research. And this document brings a table with the definition of the different types of extremophiles. Here in the first column, the factor that defines the different classes of extremophiles, here in the second column, and the defining growth condition. For example, high temperature, we call them thermophile or hyperthermophile, and so on. Low temperature, psychrophile. But what intrigues me as a biologist is the ability of organisms to resist very high levels of radiation. So the one bacterium that is very well known to do that, to survive very high doses of radiation, is Deinococcus radiodurans. And that's the bacterium that I used as a model in my PhD research in Brazil more than 10 years ago. And why the study of radiation and its effects on materials and biology is important. We know that the radiation environment in space can affect electrical components in satellites, and this can have a huge negative effect on Earth in communication satellites and so on. But what interests me as a biologist is the effect that radiation has on biological systems. And here we see a chart from the Curiosity rover on its way to Mars. So we have a radiation instrument measuring radiation through its 9 months of journey from Earth to Mars. So this is outside the Earth's magnetic field along the journey. So the radiation is much more, the radiation level is much more than what we find, for example, in the ISS, in the International Space Station. And we have a few different sources compare the radiation levels. For example, normal citizen, an annual average from all sources is exposed to 3.6 milligrays. If you do an abdominal CT scan, you are exposed to 8 milligrays, which is much more than an annual average, all sources. DOE, Department of Energy radiation worker, annual limit is set to 20 millisieverts, and astronauts on the ISS during 6 months, they're exposed to 75 millisieverts. That's a unit of radiation. And here we have 466 millisieverts of radiation. That's a lot. That's more than what is allowed by any means, by any period of time. So it's very, very high risk to go to set up a human mission to Mars at the present, because you can say that you can shield that with a thick layer of lead, for example, but it's very expensive, very heavy. There are different ideas like water, organic materials, all of this can absorb very well radiation, but there are some types of radiation that you simply cannot stop. If you think about the astronauts in the ISS, even the only Brazilian astronaut, Marcos Pontes, who is now the Minister of Science here in Brazil, he spent only 10 days in the International Space Station, and he reported that every night when he was going to sleep and close his eyes, he saw the common effect of radiation interacting with his eyes and producing that brightness. So there are some types of radiation that you simply cannot stop. Fortunately, we can learn a lot with other types of organisms that can cope with radiation really, really well. So really, the radiation in space, they come in different forms. There are different types of radiation. There is the electromagnetic radiation, there's the particle radiation, and they have different energies as well, and they come from different sources. And if you are outside the Earth's magnetic field, you are more unprotected against these kinds of radiation. So I'm going to start presenting some data from my PhD, and then move on to my postdoc, and then talk about other projects as well. So the most abundant form of radiation in space, in terms of abundance, is actually the ultraviolet radiation. There's a lot of ultraviolet radiation. If you, of course, disconsider the visible light, which is not capable of having harmful effect to life. So ultraviolet radiation has a lot of different harmful effects. And the most damaging type of radiation is UVC, because UVC is the wavelength that is absorbed by DNA, around 260. So the germicidal lamp that we have in the lab, in the laminar flow hood, emits a peak in 254 nanometers, very close to what DNA absorbs. That's why it's called germicidal lamp. But in space, we also have vacuum ultraviolet radiation, which is even more energetic, but it's a type of radiation that is stopped in the very surface layers of any material. So what is very, very damaging in space is actually UVC, of course, UVB and UVA as well. But DNA absorbs here in this range of the electromagnetic spectrum. So in my PhD, we started to test the survival of the Deinococcus radiodurans to simulated space radiation. And we started with ultraviolet radiation. And in order to have the full spectrum of ultraviolet radiation in space, we used a synchrotron radiation source in Brazil. So we have synchrotron laboratory here, and there's a beam line that emits a continued electromagnetic radiation in the visible and ultraviolet range. And the level of radiation is actually higher than what is found in space at Earth's orbit. So we use that to simulate the effects of this type of radiation on Deinococcus radiodurans in different conditions. So here on the top left image, you can see cells of Deinococcus radiodurans on a monolayer, basically, on a very smooth surface, which is a millipore filter. We also used a carbon tape to mimic a more natural environment where they can pile up and be protected in these cracks and so on. And we also use cells with culture medium. the medium that we use them to grow. So we didn't wash them. We just dehydrated the cells with the culture medium to measure how much that culture medium could protect the cells. And here we have the results, the survival curves. We have three different samples here. We have the wild type deposited on a smooth surface. We have a very, very sharp killing rate. So here is the survival rate in a logarithmic scale. And here in the x-axis is the dose. So we have a very, very high killing rate if they are totally exposed. And we also measured the protection caused by the rough surface, which is this 7 micron curve here. We tested with the wild type and a mutant that is unable to repair DNA. And even the mutant was able to survive more than the wild type on this smooth surface. And so we demonstrated that very, very small protection is already enough to protect the cells in space in terms of vacuum ultraviolet radiation. And this is published was published in 2010 in this citation here. But of course, in space, as I said, there's different types of radiation. That's not only ultraviolet. Another very abundant form of radiation in space is actually the solar wind. The solar wind is a particle radiation that is composed basically by 96% protons and electrons and 4% alpha particles on the energy of the level of 3 kilo-electron volts. That's basically the composition and energy levels of a solar wind. And this is basically mostly protected by the magnetic field. And of course, along the year or along larger time scale, this composition varies. The energy also vary with the solar energetic events. And this can go very, very high, be multiplied by thousands of times here. But this is what is constant. And this is what we try to simulate in the lab. Let me see, there's a question here. And I invite you to write, guys. Okay, yes, you can just write. I can just stop if needed. So we try to simulate these conditions in the lab using different sources. So this is also part of my PhD. We simulated first here at the top 200, oh, sorry, just hide these. Hide video panel? No, if I do that, I will never be able to come back. Sorry. So here at the top, we simulated 200 kilo-electron volt protons in, this was in Catania in collaboration with Professor Stratzula. So we used a linear accelerator to obtain these protons of this energy. This was simulating, a slightly higher peak emission of the solar wind. And we mixed the cells with different dust grains. NC means naked cells, SST is sandstone, B is basalt, and G2SiO4 is, I think it's forsterite. It's a type of meteorite. It's a type of mineral that is present in meteorites. So we mix the cells with these components to see if there is any differential effect for these types of radiation. And we observed a killing rate for the naked cells. And then cells protected either in sandstone or basalt had the same killing rate. And even the forsterite didn't provide any protection for the cells. And the doses tested here were equivalent to 100 years in space, considering this level of radiation, 200 keV particle radiation. Solar wind, the best place to whale mine helium and hydrogen, yes. And this is the Monte Carlo simulation of what is happening with the particles when they traverse a cell. The cell is represented by this orange circle. So with this energy, the particles are able to completely traverse the cell and produce a lot of different effects. That's why they are killed by this radiation. But if you use a more typical particle radiation from the sun, like 2 keV electrons and 4 keV carbon ions, the maximum dose tested for the same conditions equivalent to 1,000 years in space for 2 keV electrons and 20 years in space for 4 keV carbon ions, they simply cannot be killed. And why is that? The Monte Carlo simulation shows what happens here. This energy of particles, they cannot even penetrate the cell wall of a cell. So it's very, very weak in terms of the capacity to kill the cells. So if the cells are able to dehydrate and be there with minimal protection from UV, they can resist a lot of years in space with minimal protection. That's what we demonstrated with Deinococcus rhododendrons with these different experiments here. That makes the case to the lithopanspermia hypothesis, which is many different types of experiments have demonstrated the feasibility of these hypotheses, but we will never know if it actually happened until we find more evidence, direct evidence of that. But it's a possible mechanism, as demonstrated by different types of experiments. But what happens with the radiation when it interacts with the cell? So there are basically two types of effects. There are the direct effects that is caused on DNA. So the radiation is capable of directly interacting with DNA, causing DNA damage. And then this DNA damage needs to be restored. And if it's not stored, there's a change of genetic information that can lead to cell death or mutation. The other type of effect is the indirect effect, which is the effect of radiation on other molecules and water. Water is the main component of metabolizing cell. So there's a lot of water, and this radiation can produce a lot of different free radicals, hydroxyl, a proton, oxides of nitrogen, and superoxide, and hydrogen peroxide, and so on. All of these can have different effects on DNA, on other molecules, and also on enzymes and membrane that can lead to change of genetic information and also mutation. And all of this is influenced by the environmental conditions, mainly oxygen concentration, temperature, and pH. In the case of radiation-resistant cells, this is basically the same type of chart. It just looks intimidating, but I've tried to break down here. Here on the top, we have a sensitive cell, and here at the bottom, a resistant cell. So what is the difference? What makes a cell resistant compared to a sensitive cell? So when radiation interacts with a cell, there's indirect damage, mostly interaction with water, producing a lot of different free radicals. The hydroxyl is uniformly distributed, produce uniformly distributed cellular damage. Hydrogen peroxide and superoxide have site-specific protein damage that can lead to compromising the function of cells, including DNA repair, and then the cell leading to cell death. In the case of resistant cells, they prevent the effects of free radicals on proteins, on enzymes, mainly the DNA repair functions. And then even though the cells sense a lot of the direct effects on DNA, the DNA can be restored. For example, Deinococcus rhododendrons can have its DNA shattered up to 2,000 different fragments, and they restore these fragments within 4 hours in ideal conditions. And it's amazing, very efficient DNA repair. But it's not that they don't sense the effects. They sense the effect, but they are able to restore because they keep their proteins active, because they have these antioxidant metabolite complexes of manganese. So this was discovered, was first published in 2004 by the group of Michael Daly. And there's a lot of different evidence showing that the importance of the antioxidant metabolite complexes based on manganese on the survival of radiation-resistant cells. And here are the types of complexes that manganese do with different metabolites in the cells: orthophosphate, nucleoside analogs, amino acids, and peptides. So it basically binds or interacts with these compounds and act scavenging the effects of oxidative stress. What is the probability of a mutation being non-lethal or neutral in comparison to lethal or harmful mutation? Well, this is a general question. I would say that non-lethal or neutral mutation is more frequent than a lethal or harmful mutation, because you never know what changed until you see a phenotype. And let me see, let me think about it. A mutation is only, you can only talk about mutation if the change of genetic information is passed through generations. So that means that that change was fixed in the population. So if it was fixed, it was not that harmful, right? But if you talk about change of genetic information caused by radiation, then it depends on the dose, right? It all depends on the dose. People who study the effects of radiation at the very, very fine tuning, you know, when the cell starts to sense, they work actually with very, very small doses of radiation, and then you can study and you can say if a change of genetic information is non-lethal or neutral. But I would say that non-lethal or neutral mutation is more frequent than lethal or harmful. I don't know. I would have to think more about that. Let me see. Yes, and one correlation that was established between radiation resistance and dehydration resistance is microorganisms or organisms that resist to dehydration, lack of water, they tend to be also radiation resistant. Because if you think about a cell undergoing dehydration, you can imagine the physical alteration of the cell. So here we have a cell very turgid, very happy in a water environment. When it undergoes dehydration, it undergoes biochemical stress, so a lot of accumulation of reactive oxygen species, metabolic stress, physical stress, and physiological stress. So you can simply imagine, you know, like a cell wilting, losing water, and that causes a physical stress that can lead to breakage of the DNA molecule, of impacting many different types of the cellular components, but the most important ones are the ones that carry information, which is the DNA. And then DNA can suffer, you know, single strand and double strand breaks, which is the type of effect that is caused by the direct effects of radiation, of ionizing radiation. So for my postdoc, we asked the question, is Deinococcus radiodurans the last frontier of radiation resistance in nature? And we hypothesized that other organisms could accumulate higher levels of manganese, they could have more efficient DNA repair tool set, and could produce unusual antioxidant secondary metabolites. We are actually still on this search. You know, as I said, as we mentioned earlier, I've just been to Saudi Arabia to do more research on radiation-resistant microorganisms from deserts. Just because, you know, nature is there and we didn't find anything more resistant than Deinococcus radiodurans yet, but we found some very interesting microorganisms that I'm going to show now to you. So we set off to field expedition to the Atacama Desert, which is the driest place on Earth. It's considered a Mars analog because of the dehydration. You can see here a landscape. There's no vegetation, very few clouds there. There are reports of sites there that didn't see any drop of water in the last many decades and so on. Very, very out of this world landscape. And here's me collecting samples there at the Atacama. We also collected samples in the Sonoran Desert in Arizona. And we had a film crew from the NHK television in Japan following for, you know, for an episode of the Cosmic Front documentary, which was very nice. The procedure after collecting samples is basically exposing these sand samples to a source of UVC. Actually, we first sprinkled these sand samples over solidified culture medium. And then after exposure of, to 300 joules per square meter, which is considered very, very high for most organisms except Deinococcus, we were able to isolate many different types of morphotypes, which are different types of microorganisms growing there on these agar plates. And then we selected the organisms and classified them by their level of resistance compared to Deinococcus radiodurans here at the top. So here's a chart listing all the different isolates and the survival fraction, the survival level to 300 joules per square meter. And as you can see, we have a gradient of different organisms showing different levels of radiation resistance. So we have a microbial collection there at NASA of organisms from deserts that have different radiation resistant profiles. And here on the left, we have a chart just comparing the biodiversity of isolates of organisms from these different places. Here, the first bar is the Ames Research Center, our control sample. MN is a manganese mine in Sonoran Desert in the USA. Here's the Atacama Desert. You can see that the biodiversity, each color here represents a group of organisms. So you can see that the Atacama Desert is the most diverse environment that provides the most diverse types of organisms. And F and S are different types, different methodologies of isolating these organisms. F is filtration, when you dilute these sand samples in a saline or a buffer. And S is the sprinkling method, when you don't interfere with the microbial community, you just sprinkle them on an agar plate. That method provided the most diverse, the highest diversity of organisms compared to the filtration method. So that's what we used in our further studies. We also looked into the relationship between manganese and radiation-resistant isolates. So we measured the extracellular manganese in the samples. So we just sent out these samples to measure for a company that measured the concentration of manganese and iron, because iron also has a role in radiation sensitivity. Organisms that have high iron inside the cells, they produce a lot of reactive oxygen species through phantom reactions. So the important thing is the balance between manganese and iron. So that's why we measured manganese and iron in the sample, so extracellular versus survival. And as you can see, there's no correlation, between the concentration of manganese or iron in the sample and the survival of isolates. But if you look into the intracellular concentration of manganese and iron, you do this ratio, and then you can see a correlation. Organisms that have high manganese to iron ratio, they tend to be resistant to radiation. And those that have high iron and low manganese, they tend to be sensitive to radiation. And that's a profile that had been already demonstrated in the literature, but that's the first time that we demonstrated for environmental isolates for UVC radiation. And that corroborates with what is out there for ionizing radiation for laboratory strains. And then we measured the radiation resistance of the top two radiation resistant isolates, comparing them to Deinococcus radiodurans in blue here. So we actually found an organism called Hymenobacter that is more resistant than Deinococcus radiodurans to UVC radiation, and one called Extremophiles that is more resistant at highest doses. And I'm going to explain why this is the case. But if we do the same survival curve using other type of radiation, which is a very high energy ionizing radiation, then Deinococcus is still the champion. Hymenobacter is the second one, and Extremophiles is the third one. But even Hymenobacter and Extremophiles, they have very high survival profile. This dose here, 5 kilograms, is actually 1,000 times higher than what a human cell can tolerate. So very, very good models for further studies of radiation resistance. And here we have on the left, the Hymenobacter isolate. We can see that they don't form cell clumps, and it's not possible to see here, but they don't produce a very strong pigment. They produce a pigment, but it's very light pink pigment. So probably the pigment is not doing much in protecting against radiation. And we hypothesize that the main thing going on here is the DNA repair. which is a very good model for further studies of the molecular mechanisms involved in radiation resistance. And here is the Geodermatophilus isolate, the one that had that broken curve that is more resistant than the coccus to the highest radiation. That's because they produce cell clumps and they produce very strong dark pigment that absorbs a lot of this type of radiation. And so they are killed at the starting doses, but then the ones that are protected within the cell clumps, they survive and they show up at the highest doses. So we, yeah. Sorry, is that a question?
No,
I
it's
think
not.
someone, okay, no problem. So we selected the two groups that have the most different types of radiation-resistant organisms, which are the Arthrobacter and Cocuria. Both of them are actinobacteria. And we had, I think, 12 samples of them, 12 replicates of them. And in a collaboration with Dr. Kazuharu Arakawa from the Institute of Advanced Biosciences in Japan, we repeated these experiments and in a very controlled manner. And we are sequencing the whole genome sequencing of each one of them to establish exactly side by side what is the genomic difference between them that makes them have this gradient of radiation resistance. So this is an ongoing work. These organisms are highlighted here in the first chart that I showed, listing all the different types of organisms. And so we expect to have very interesting results from this collaboration. First few papers are starting to be published, like this one, the genome sequence of Arthrobacter, one of these strains. And all of this is related to the exploration of space, which is the third question of astrobiology, which is what is the future of life? As you know, astrobiology has these three questions, the origin of life, how did we come here? Where did you come from? And then what is the possibility of finding life elsewhere? And then the future of life. And all of this research has the potential to impact future exploration of space because synthetic biology, is improving the performance of organisms in different industries here on Earth. And all of these will be exported to other places beyond Earth because we need them to explore. We cannot bring all the resources that we need in terms of water, food, and everything that we will need in a long-term human space mission. And if we think about it, cyanobacteria has a very, very high potential of helping space exploration because they can be cultivated with minimal energy because they only need solar radiation, energy, and heat. CO2, some of them can also fix nitrogen. They can use water, which is present, for example, on Mars and even the moon. And they need some micronutrients that are already there. And they produce organics and other materials to grow heterotrophs. And all of these can produce food, clothes, and materials that will be needed. So cyanobacteria has a very, very high potential to help space exploration. And based on this idea, in 2011, our first iGEM team came up with this idea to genetically engineer a cyanobacterium called Anabena to produce and secrete sucrose, a sugar that could be used by another organism, Bacillus subtilis, which could then produce food, fuel, clothes, drugs, bricks, and so on. So they did that in 2011. They were very successful, won several awards. And then in 2013, we received a visit from representatives of the German Space Agency, DLR, and they invited us to fly a payload on their satellite called EUCROPIS, which is the acronym for Euglena, Combined Regenerative Organic Food Production in Space. So they would have their own experiment growing another type of organism called Euglena. They had their own experiment and we would be the secondary payload. And since then, we started to work on this EUCROPIS satellite mission to develop this secondary payload with our own experiments. So the name of our payload is PowerCell, which would demonstrate the concept of one organism providing energy to grow another organism. So that's why it's called PowerCell. And then that paves the way for human exploration of Mars and Moon. We also designed a transformation experiment to happen in space. That's the first time transformation happened in space. That paved the way for symbiotechnology in space. We also had two more experiments, protein production experiment based on the beta-glucuronidase report system, paves the way for bioproduction on Earth. I'm gonna talk more about each one of these experiments in a minute. And growth in LB, which is our positive control, basically how they grow in ideal conditions, demonstrate the biotechnology reliability in space and paves the way for microbiology in space. All of this in a microfluidic card that fits in the palm of a hand. I actually have a unit here. So this is the size of a microfluidic card. So it's a very tiny card with 48 wells. So 4 rows of 12 wells, each one containing 70 microliters of volume capacity. So we had different experiments in these microfluidic card. This is a cross-section of a microfluidic card. It's a drawing, a schematic drawing, showing that we had an LED board with light sources that would go through these wells. And at the bottom, we had a detector chip to measure the amount of light that had been absorbed, that is being absorbed by culture medium, by cells growing in the culture medium after we activate the experiment. And the more the cells grow, the more they absorb light, the less light will reach the detector. And that's how we did all the experiments. Basically, it's a miniaturized spectrophotometer in space. We originally had different lights, different wavelengths for these LEDs because we changed many times the experiments and the experimental design, but that's what we had in our hardware. And here is a schematic of our experiments, how they were distributed in these microfluidic cards. So we had basically these 4 experiments here. The X-gluc is the protein production experiment. Lysate extract is the power cell. LB control is our positive control. And this is the transformation experiments. And I numbered them here so that you can keep that in mind. So number 1 is transformation. LB is number 2. Lysate extracts is number 3. And X-gluc is the protein production is number 4. So we had these valves and all these mechanics controlling the activation of these experiments, and a waste bag to receive all the fluids after the experiments have been done. So we had actually 2 payload enclosures, A and B, each one with 2 microfluidic cards, so we could repeat all the experiments in different conditions. And these conditions are actually different gravity regimens. Because the satellite is a platform that can control the spinning of the satellite to simulate the gravity of Mars, Moon, and microgravity just by changing the speed of their spinning. And so we could repeat all the experiments in different conditions. So all of these were fit into the payload module. So we had 4 payload modules within 2 payload enclosures. So here is a more detailed explanation. So we have these microfluidic cards. A payload module included a LED board and also a thermal spreader to keep the temperature at 30 degrees roughly. And this is a payload assembly. So we have here payload enclosure A and payload enclosure B at opposite sides of the satellite, the solar panels and so on. So they rotate and they simulate by centrifugal force the artificial gravity compatible to what is found on the Moon and on Mars. So we have this axis, which is pointing to the sun. So it's a sun synchronous orbit. And we were able to control the spinning of the satellite. And here's a photograph showing the solar panels deployment and our payload here. The other one is the opposite side and we cannot see. What is your opinion on industry farming, the use of tailored organism bio-based robots like nanobots and microbots for making celestial body better for human or robotic use, such as mining, manufacture, development, settlement. I think I'm going to comment that at the end of my presentation. Thank you for the question, Camille. And then we have the vibration test. So after we assemble everything and ship to Germany, everything needs to survive a vibration test, because that's what the experiment will experience during launch. And if it fails, we had to repeat everything, figure out what failed, and just repeat. So of course, when it passes, everyone is happy, even the Germans. They were very excited that everything worked as planned. And since this was a partnership between NASA and the German Aerospace Agency, we had to prepare everything at NASA and then ship everything to Germany. Everything were integrated there. And then this satellite came back to the US and was launched from the Vandenberg Space Base on December 3rd, 2018. And Tomasz went there with me, and we both watched the launch live, which was a very, very exciting experience. Here are the logos of the mission, the logo from the DLR, German Space Agency, our logo, the power cell representing Mars, Moon, and a petri dish with cyanobacteria here, and a symbol of energy, representing the cyanobacteria providing energy for other organisms. Here is the logo from the space flight company that managed the launch of 64 satellites, including a Brazilian satellite. I think it was the first Brazilian satellite developed in Brazil, in a university, that we're able to fly there as well. And here's the logo from SpaceX. And they'll always have this green plant here to provide luck. And here's a short video of the mission. It's like 1 minute video that was produced by Blue Marble, which I think is awesome, and shows a different aspect of the mission, which is the potential of these results that we are getting now for in situ resource utilization. Because if we demonstrate that the cells, the behavior of the cells in different worlds, we can, make the point that it will work in other planets.
Stage 2 is pressing play. Launch will track on that one. We'll go for launch.
Okay, I hope it was not too loud. So we have I broke down the results by the different experiments. The first one is the genetic transformation, the results from the genetic transformation. We have here very good results of transformation in microgravity, and to our knowledge is the first time an OGM, GMO, genetic modified organism, was produced in space. by an automated procedure. Everything was automated. So we have here on the orange line, the growth profile of our positive control, the neomycin resistant strain. So we included antibiotics. So only the organisms that resisted that had the resistant gene were able to grow. And we had 2 replicates out of 5 replicates that were able to grow, just like very similar to our positive control. And we have here the negative control with the baseline, the blank. So very, very beautiful result that indicated the transformation in space in microgravity. That was our first experiment in space, the first one that we activated. Here we have the subtitles. We have the different strains that we used. Strain 168 is our wild type. 1A976 is the parental strains, our negative control. 1A976 neo R is the positive control. PVEG is for the protein production. 1A976 under a very strong promoter for protein production, and P-LIAG weak promoter, and 1A976T is the transforming strain, the strain that was supposed to receive the gene that conferred the resistance to neomycin, to this antibiotic. And that's a very, very good result. So as I said, the baseline, with the negative control with the baseline, and then our positive control, and 2 replicates of our test samples. Conclusion, although not all replicates have grown as desirable, only 2 out of 5, the results are compatible with a transformation scenario, indicating that the transformation experiment has worked in microgravity. So we were very excited about this result in microgravity. That didn't repeat in the other conditions, as I'm going to show later. Here we also had transformation in our ground control, but only 1 replicate behaved like our positive control. We have here replicates 1 and 2 for the positive control, and only 1 replicate out of 5 that grew like the positive control. And here we have negative control. Here I removed the baseline here. So there were some problems in different experiments with the hardware. So only the results that were very indicative of what we expected, we considered successful. So the conclusion here is that considering growth above the negative controls as an indicator of successful transformation has worked for that particular replicate. So only 1 out of 5, but still, it's a transforming replicate, just like the positive control and the negative control here. So we had some unexpected result. So we had a replicate of all the different experiments for all the different conditions in our lab as a ground control. And there we were able to open the unit and see what was going on. And we noticed for the critical samples for transformation and also for our control in LB, we used LB with a component to induce genetic transformation, which is xylose. And xylose at high concentration, 2%, over a very long period of time, they undergo what is called Maillard reaction. And so they precipitate and they form a type of nutrient that is not usable for the microorganism. So we think we were very lucky that the first experiment worked, because it was not enough time for that Maillard reaction to affect the results. But this is what we observed. We measured, we did some chemical analysis, and that's our conclusions. Maillard reaction going on and preventing the growth of the organisms, except the first one that we did for transformation. The second experiment is the growth in LB, which was the most beautiful result. We observed growth in the first few days of growth for both strains that we were testing, 168 and 1A976. Very, very beautiful growth profiles for the lunar condition. And then number 3 is growth in lysate. So the growth is very, very low. This was actually expected because we didn't use the cyanobacteria itself. We had to use cell extract and then pass that extract through the filters. So that limited the amount of carbon for them to grow. But this was expected, and we didn't see any difference between the two strains, and we actually were successful growing them in cyanobacterial cell lysate. And this is the protein production. For those of you who are more familiar with molecular biology, we use this report system based on beta-glucuronidase. It's an enzyme that degrades complex carbohydrates. And there's an analog of their substrate called X-gluc. This X-gluc, when broken down, they produce a bright color in the medium. They turn the color of the medium into blue. And this is our laboratory results, what we expected to see in the mission. So the strong promoter would produce more of this protein, would degrade more substrate, and would be brighter blue. And the other one would still be blue, but very weak. And this is our negative control. We have results here. So the NEOR, the P-LIG and the P-VEG up there, the P-LIG, which is the weak promoter here, and the negative control there. This is what we expected to see in the mission, but it didn't work as planned. I would like to ask, okay, I'm gonna read the questions later because I'm late in my presentation. Let me just explain why did we didn't see this in the mission. So this is what we saw. We only saw growth of the different strains that we use, the NEOR, the P-VEG, and P-LIG, because this component that we use, the X-gluc, it degrades after a few weeks on liquid form at room temperature. It was actually, it was a last minute addition to the mission design. We had to load the samples within a few months. So we selected this experiment, but we didn't have time to test the degradation of this component after many, many weeks. And we had to wait 2 years to fly this mission. So of course, we later found out that it degrades after a few weeks. So we were unable to measure protein production, but we saw the best growth curves for these experiments if we don't consider the X-gluc, because the X-gluc degrades but didn't interfere in the growth. So we had the best growth curves for all different conditions, as I'm gonna show next. So now I'm breaking down by the different gravity levels, so compare the different gravity by the different strains. So here's the transformation experiment, the different strains here. So we see that the different effects of microgravity, lunar, Martian, and ground throughout all the next charts. I'm going to use the same color coding. So microgravity, we didn't see any difference between microgravity and ground controls for the transformation for this particular strain, but we can't say much about lunar and Mars. The Mars experiment, actually, the experiment that were activated under a Mars simulated gravity, that particular payload had a lot of problems. So we didn't observe much growth for all the different types of experiments, for all the different strains. So we can't say much about that. And this is the other strain, 1A976. For the lunar experiment, we see growth very well for all the different strains for the transformation experiments. And then the ground control was kind of finicky. It showed growth at middle point, so we can't say much the effects of gravity for the different strains for the transformation experiment. We only had that result that transformation worked, but it's difficult to do the comparison between the different gravity regimens for the transformation experiment, because it only worked for the microgravity condition. And here we have growth in LB, that's our positive control, that worked best for the lunar condition, for the moon gravity, for the two strains that we tested, 168 and 1A976. All the other conditions, we can't say much. It didn't work very well because this experiment had that xylose that interfered with growth. The lunar condition was our second one, and maybe that's why it did grow very well. It didn't interfere much. Then we did the Martian, and then we did one repetition for the Martian. This is our growth in lysate that worked very well for both strains 168 and 18976. This is an anomaly, of course. There's not such concentration of carbon to produce this amount of growth, but this is just to show you that there is an anomaly that happened for the microgravity experiments after 40 days of growth. But as you can see, they were all able to grow in lysate, and there's no difference between the different types of gravity regimens. And this is the fourth experiment, our X-globe control. As I said, the best growth curves were obtained for this experiment, and we didn't see any significant difference between the different gravity regimens, regardless of the type of strain. This indicates that biology can behave as expected in different worlds, that the gravity doesn't have much effect on the growth of organisms when we consider a long-term experiment like many days. Of course, we can fine-tune that in future experiments to see what is causing this little behavior, if this is significant or not. But looking at the data here, they overlap in different times. So it doesn't allow us to say anything about any difference between the different gravity regimens for these experiments for these strains. So we have over 400 days worth of mission data, being 50% flight data and 50% ground data. We did observe growth in all 4 gravity regimens, one way or another. We did successful genetic transformation in microgravity, successful growth of bacillus cyanobacterial cell extract in 3 out of 4 gravity regimens. We had a problem with the Martian, the 4th experiment in the Martian condition. All hardware and software working as planned, mostly. Of course, we had problems in a few experiments. Demonstrated ability of hardy bacterial spores to recover from a long-term stasis period, which will be fundamental for human deep space missions. Valuable long-term reagent stability data, including antibiotic efficacy. We actually had almost 5 years worth of data indicating the efficacy of neomycin antibiotic, which is a huge finding, because if we think about it, all these different biological compounds, the companies, they only test like 6 months or 1 year. They don't have any intention to test more because it costs, and it's not supposed for that long period on the shelf. But if you think about a space mission, that's very, very important to see how much they can be stable. And we demonstrated, the experiment is still going on, we still have many time points to analyze for the neomycin antibiotics. And then this raises the question, what other biological compounds would resist and be stable for many, many years? That this could save a lot of money when planning a human long-term space mission. So we had numerous lessons learned from hardware design to experimental operations that are leading to improved designs and capabilities for future missions. And this is just to show the criteria. I showed a lot of unexpected and negative results and other positive results, but this is how you design a mission. You design a mission, you set up the success criteria, so the minimum success criteria, full success criteria, and extended full success criteria. And even with all these negative results, we were able to reach a complete status for all of them, because this was mostly a technology demonstration mission. So the scientific, which we think as a scientist is more important aspect, was not the main point of this mission. If it was, we would have a better design and better experiments, more time to test, and so on. But in terms of technology, we were able to reach extended full success. And the only thing now being left is submission of papers for publication in peer-reviewed journals. which we are already doing. And so we expect to see some papers this year. And we are reaching the end of the presentation. And I just would like to mention this Young Scientist program from Blue Marble that's gonna open, I think in February. So the Young Scientist program, it provides opportunities for students and early career scientists all over the world to collaborate with PIs from Blue Marble. Many of them have research in collaboration with NASA. And it's an opportunity for them to develop research during a 3-month program. It's a summer program. And they attend events and training, public speaking, outreach, and ethics, and so on. So very, very good opportunity. They receive a certificate in the end and have the possibility of publishing papers and abstracts to conference with Blue Marble PIs on NASA projects. Here's just to show how many applications we had from all over the world: 325 applications in 2021, 128 applications accepted after a peer review process. And yellow here on the left is only the 2020 applicants, and red is 2021 applications. And the residing locations of 2021 Blue Marble Space Institute of Science Young Science Program Research Associates. So they become research associates when they are accepted to this program. So keep an eye in our website here for Young Scientist program. And the application is going to open next month. And then I just would like to finish with this thought by Konstantin Tsiolkovsky: the Earth is the cradle of humanity, but one cannot live in a cradle forever. That doesn't mean that we are going to trash the Earth and move to another place to trash. I like to think about this problem, about this thought, looking back to history, where we had different cultures spreading all over the planet, all over Earth, but they didn't have to leave their homeland. They are still there, and we can think about the different empires, the different cultures that had spread over the Earth. That's what's going to happen in the future. We don't need to leave Earth. We just need to spread beyond Earth to increase our chances of survival to natural disasters and also artificial disasters, if we consider human nature. I think that's all. Thank you. It was a long presentation. I tried to fit in 1 hour, and it clearly it didn't happen. So if we have time, I'd like to take these questions, and if we have more questions, I'll be here available to answer them. Thank you so much.